start with the introduction of multiplex PCR and molecular diagnostic panels in infectious disease testing. Molecular diagnostics are important for rapid and accurate detection of infectious disease. Polymerase chain reaction, or PCR, is the fundamental technique of multiplex PCR, which are used in many molecular diagnostic tests. Now, let's revise a bit about PCR. PCR is an enzyme-driven method to amplify and detect genetic materials from pathogens. It uses primers, thermostable polymerase, and thermal cycling, and can produce millions of DNA copies in under an hour.
Multiplex PCR is a technique that amplifies two or more DNA targets at the same time in a single reaction tube by using multiple primer pads. It allows simultaneous detection of several sequences and is commonly used in genotyping, forensic testing, and pathogen identification. There are two types of multiplex PCR. First is single template PCR reaction, where it uses a single template, which can be a genomic DNA, along with several pairs of forward and reverse primers to amplify specific regions within a template. The second one is multiple template PCR reaction. This reaction uses multiple templates and several primer sets in the same reaction tube. Presence of multiple primer may lead to cross-hybridization with each other and the possibility of mispriming with other templates. Let's move on how multiplex PCR works. First is denaturation. The reaction is heated at 95 degrees Celsius to separate double-stranded DNA into single strands. Next is annealing, where the temperature is lowered to allow all primer sets to bind to their specific target sequences. Annealing temperature must be optimized so every primer pack can anneal efficiently at the same temperature. And the last one is extension, where the DNA polymerase will extend from each primer to generate multiple DNA fragments of different sizes simultaneously.
Now, I will give an overview of molecular diagnostic panels, also known as syndromic panels. Molecular diagnostic panels are a test that designed to detect many possible pathogens at the same time using just one patient sample. Instead of running multiple individual tests, this single panel can detect for bacteria, viruses, and other organisms that causes similar symptoms. A good example is respiratory panels. It can detect about 20 to 25 pathogens, including common viruses such as influenza A and B, adenovirus, and SARS-CoV-2, as well as bacterial pathogens such as Mycoplasma pneumoniae and Bordetella pertussis. There are also gastrointestinal panels, which detect around 15 to 20 pathogens, including bacteria like Shigella and Salmonella, viruses like rotavirus, and parasites such as Giardia. Another important test is blood culture identification. This test is used when blood culture is positive and rapidly tests to identify whether gram-positive or gram-negative bacteria, yeast, and key resistant genes such as MAC-A, VAN-B, and VAN-A. We can classify diagnostic panels into two main ways. First, by clinical syndromes, which include respiratory panels, gastrointestinal panels, sepsis or bloodstream infection panels, and meningitis panels. Second, we can classify it by technology or platform use. For examples, multiplex PCR, microarray-based system, and lastly, isothermal amplification method.
Now, let's move to the advantages and limitations of multiplex PCR in molecular diagnostic panels. For the advantages, multiplex PCR can detect multiple pathogens or targets in a single reaction. It also provides high reproducibility and standardization, offers faster turnaround time compared to running multiple single PCRs, has high sensitivity and specificity, and saves time, reagents, and sample volume. For the limitations, too many targets can cause competition between primers. Other than that, some panels only detect organisms included in the panel, so rare or unexpected pathogens can be missed. Expensive instruments and reagent cartridges can restrict accessibility and increase per test cost. Also, it often cannot distinguish colonization from true infection, and requires skilled personnel for optimization. Now, let's dig into the clinical case using multiplex PCR and diagnostic panels. This research was done to study on how effective multiplex PCR technique specifically for BioFire field array pneumonia panel. This clinical study involved 140 immunocompromised patients with severe pneumonia in ICU. The respiratory specimen was taken and processed with multiplex PCR and also conventional culture. The main comparison between these two is the time taken for the results to be obtained. Culture will take 2 to 3 days to determine the responsible pathogen, while multiplex PCR only take 4 hours. Multiplex PCR provides results with high sensitivity, specificity, and also high negative predictive value. This is very important to rule out major pathogens such as Pseudomonas and MRSA. Based on the result from the research, it detected mostly Enterobacteriaceae, Haemophilus influenzae, and also Staphylococcus aureus. This fast and advanced technique helps change antibiotic therapy in 17.5% patients. These bar charts compare the pathogens detected by BioFire field array pneumonia panel shown in black versus conventional culture shown in grey. Multiplex PCR detects key respiratory pathogens at higher rates than culture, but culture still identifies many organisms that are not covered on PCR panel, showing both methods are complementary. In conclusion, multiplex PCR and molecular diagnostic panels offer high sensitivity and excellent negative predictive value. However, despite its strength, multiplex PCR cannot detect organisms outside the panel, so culture remains essential. Overall, multiplex PCR and molecular diagnostic panels represent a major advancements in modern microbiology, providing a faster and clinically targeted results.